Hello students, welcome back to NEET World channel. This is Narayani, I am your botany educator. So in previous class, we studied the very important question for IPE board students, that is nodule formation, right? So before we revise that topic, so let us tell you very important news here all of you. The new batches already has been started students. So the current batches are also running. So the next batch is going to start from the September 1st onwards students. So we are giving this NEET program especially for very affordable price at just rupees 3,333 per month. Yes, you heard it right. If you are really passionate about your dream to become a doctor, so this is the best platform for you. It is especially very best opportunity for the 11th and 12th goers who are going to college, because if you are very smart in preparation, you can cover both IPE as well as the with us NEET coaching. So as it is very affordable fee students, it is quite good opportunity for you. So it will get you good rank in your upcoming competitive examination like NEET and EAP set examinations. So it is available both offline as well as online students. So if you prefer online, we will access in our app name called NEET World. So if you want to join through offline, you can come to our institute name called NEET World, present in Narayani, Hyderabad. If you are 12th completed students also, we have long-term batch students. So you can join with us as per your convenience of both offline as well as online. So don't miss this opportunity. Just this course is providing just rupees 3,333 per month along with study material students. If all of you go through our live plus recording sessions, and you do our well crafted study material, it's enough to secure good rank in an upcoming examination for sure. Okay, so don't let this opportunity go away from your hands, students. So another more very important news is, so this is our Telegram channel students. By scanning this QR code, you can access our Telegram channel. For any competitive examinations, doing mocks is very important. So daily we are posting MCQs of biology, chemistry, physics, mathematics and also chemistry. So every subject we are posting the mocks right there. So thereby it can help your topics. Based on the topics we are providing the MCQs. So it will help you to boost your confidence, as well as it will help you crack the how to application based questions to do and all that. And one more important thing students, the live classes pre scheduled will be informed there, though thereby you can join in our live classes. And also what are the free resources, digitalized materials, handwritten notes will directly provide in our Telegram channel. And also if you have any doubts, you can post there by tagging the particular lecturer, so thereby they can clear you all your doubts for sure. So if you have any further more queries, please contact this number. We will therefore help you out all your doubts to clear off. Okay. So now students, in yesterday's session we discussed how the nodule is formed, right? So what is the main important for the nodule formation students? That is nitrogenase enzyme. Nitrogenase enzyme. For the biological fixation of nitrogen, the nitrogen is going to converted into ammonia or ammonium, right? So as for the conversation, which organisms are helping out? That are prokaryotic organisms. Why those only can fix the nitrogen students? Because they have the specialized enzyme name called nitrogen present in their prokaryotic living organisms. They have the very important enzyme name called nitrogenase. So this nitrogenase can fix the nitrogen. So by a symbiotic association with the plants, they also fixing the nitrogen. So during that process they are forming the nodule, right? They are forming the nodule. So mostly the rhizobium bacteria, which is in the rod shaped bacteria, mostly it forms the nodules in the leguminaceae plants. So very important students, examples are also very important. So such as alfalfa, sweet clover, sweet pea, lentils, garden pea, broad bean, clover beans, etc. So remember these as per your NCERT book, students. So these are the must and should examples you have to remember for sure. And some other bacteria like Frankia, it is associated, it is also shows symbiotic association with non-leguminous plant, that is Alanus. That is Alanus. So now see how the nodule is formed, students. So it is not a obligatory, it is not mandatory process, right? So whenever the the plant required nitrogen, what will the plant will do? The root hair releases the chemoattractants. The root hair releases the chemoattractants. So the bacteria present in it are get attracted to that and attach it to their receptors called lectin receptors. What are the chemoattractants, students? That are flavonoids and betaines, right? Flavonoids and betaines. Is it visible? Okay. So see, this here the chemoattractants released by the root hair, and the bacteria got attracted and attached to its receptors called lectin receptors. Through that, so the root hair forming the curl and the bacteria is invading into this epidermal root hair, right? So through the epidermis it reaches to the cortex and forms the and leads to differentiated the its content, right? The bacteria get differentiated. When the bacteria is invading, it loses its hard cell wall and it is covered with the plant derived structures, and when they are differentiated they form the mature nodule. What is the work of nodule, students? That is, it blocks the oxygen. What will it do? What is the work it will do? It blocks the oxygen. Why to need to block the oxygen? Because the nitrogenase enzyme is specially active in the absence of oxygen only. So when it is blocking the oxygen, the nitrogenase enzyme fixes the nitrogen. So the nitrogen components are very much required for the plants, that to prepare the proteins, amino acids, nucleic acids, right? So now see, students, so till now we are discussed the nodule formation, right? So now see, what is the why we call it a symbiotic relationship, students? The host is getting benefited by the nitrogen element, and coming to the bacteria, how it is benefiting? It is directly connecting to its vascular system, so thereby it is exchanging the nutrients and it is also getting the shelter from the plant, right? So there is a mutual benefit is happening, that is why it is a symbiosis process. Okay. So now see, is that nitrogen, how it is fixing? It is directly fixing the nitrogen in one stem? No, not like that. If this is the nitrogenase enzyme. This is the nitrogenase enzyme. So when there is absence of oxygen, this nitrogenase enzyme get activated. Okay. So when it is activated, it is able to fix the nitrogen, right? Which means atmospheric nitrogen, right? So atmospheric nitrogen is in the form of N2, right? It is in the form of N2. So when it is not directly, when directly the nitrogen is not converted into ammonia, it takes 3 steps process. It takes, students, 3 steps process. That is, see, this nitrogen first it is reduced into NH double bond NH, NH double bond NH. So next, this is the first step. So coming to the next step, this NH double bond NH again get reduced into NH2 single bond NH2. Okay, wait 1 minute, students. So NH2 single bond NH2. Okay. So now see, this NH2 now again get converted into 2NH3. So directly the nitrogen is not forming. It takes the series of steps, that are 3 steps. For each step requires the 2 electrons. Each step requires 2 electrons. So how many electrons are there? Total 6 electrons are required to convert the nitrogenous, to convert the ammonia, right? But so to attach this nitrogen is also, there is a requirement of electron of 2 electrons are required, which means total 6 plus 2, 8 electrons required. How many electrons required, students? 6 plus 2, 8 electrons are required to complete the 3 steps process. Got it? Now see, this nitrogen enzyme fixing the first nitrogen into this NH, right? So this is the NH double bond, NH double bond, NH, right? Got it? So this, when it is reducing here into, 1 minute students, this again reduced into NH2, NH2, right? So this again NH2 get reduced into the third step, NH3, NH3. We all know that NH3 is the ammonia, right? So now what will happen? So to make the charge balance, because here it is required to complete these 3 steps, there is how many electrons are required, students? There are 8 electrons are required. So to make the charge balance, what will happen? There is a involvement of 8 H plus ions also to make the charge balance. So when for 8 electrons, there is a release of, for each electron there is a release of 2 ATP. How many ATP required for 2 electrons? 2 ATP are required. So here 8 electrons are required. How many electrons, students? 8 electrons are required. So for 8 electrons, how many ATP are going to involve in the process, students? There are, for each electron there is a requirement of 2 electrons, right? So 8 to the 16 ATP is required. 8 to the 16 ATP are required. Got it? For each electron, how many ATPs are required, students? There is a 2 ATPs are required. 8 to the 16 ATP are required to complete this process, right? So there is a 16 ATP. They are hydrolyzed to form 16 ADP plus 16 Pi. 16 Pi. So this nitrogen is getting converted into ammonia. This nitrogen is getting converted into ammonia. So, and this C, this H plus ions are getting converted into H2. Got it? So ultimately the nitrogen is converting into ammonia. The nitrogen is getting converted into ammonia. Okay? So this is how the nitrogen fixation is happening in the plants. Got it, students? How many steps are involving to complete this, to form the nitrogen into ammonia, students? There are 3 steps are involved. For each step there is a requirement of 2 electrons. So to make the charge balance, there will be a 2 H plus ions are also required. For to the fixation of nitrogen also, electron is required. There is the who donates that electron? Ferredoxin. Ferredoxin is the electron donor for the nitrogen. Who is the donor of the electron donor for the nitrogen fixation, students? That is ferredoxin. So including that, it is the 8 electrons are produced. For each electron, how many ATPs are required, students? 2 ATPs are required. For 2 electrons, there is a requirement of 2 electrons are required. 2 ATPs are required. For 8 electrons, there is a requirement of, for each one is having the 2, 8 to the 16 ATP are required, right? For each one, there is a requirement of 2, that is why 16 ATP are required. So to produce this ammonia. So when the conversion is happening, so 16 ATP are hydrolyzes into 16 ADP plus 16 Pi in organic phosphate. Got it? This is how the nitrogen fixation happen in plant, students. So it is very important step, students. Okay. Now see, we learned about the nodule formation, and now we came to know about how the nitrogen is fixing, how the nitrogen is forming the NH3, right? So we all know that so the nitrogen take from the roots by the plants, right? From the roots only the nitrogen is taken up the plant. The mostly the nitrogen available in the soil in the form of nitrates, in the form of nitrates. The most available in the soil in the form of nitrates only, mostly, or ammonia, So now see, so in the soil the plant is always taken in these forms. So but we know that we don't have, plants don't have that mechanism, right? But plants can take their forms as the nitrates and ammonia forms, right? So when it is taking these nitrates or ammonia, they are very toxicities containing. So the plants cannot assimilate these products also directly. Okay, if they observed also, they are not to be, so their lead is, if they are more in amount or if they are present in the plant, they creates the toxicity. So that is why these are also not suitable forms for the to the plants and also human beings, for also animals. Then how they are stored, how the nitrogen is utilized? So these, whatever the compounds are absorbing by the plants, they are converting into amino acids. They are converting into amino acids, right? So we all know that amino acids are the building blocks of proteins, right? Amino acids are the building blocks of proteins. So for protein is required everything for us, right? In the human or plants, proteins are required. So they are converted into the useful forms like amino acids. And for these students, this is one of the most important question for the IPE boards also. Look into here. Write in brief how plants synthesize amino acids. They are asking about how plants synthesize amino acids. How to synthesize the amino acids, what is requirement element for the plants? Nitrogen, right? It is taken from the roots in the form of nitrates or ammonia, right? Mostly it takes the nitrates form. So but it is suitable to take the nitrate form, students? No, it is not at all suitable. Then what will happen? It turns into ammonia. So this ammonia forms the suitable assimilated form, which is can incorporate into their structures, that is amino acids, right? So they will convert into amino acids by the two-step process. Now see, here they are asking, right, how plants synthesize amino acids. Amino acids in plants are synthesized in two ways. They are having the two ways. One is reductive amination, another one is transamination. What are the two methods, students? Reductive amination and transamination. So now first see the reductive amination. So the word itself saying that reducing, right? Reductive amination. In this process, ammonia reacts with alpha ketogluteric acid. So we all know that when the cellular respiration is happening, the alpha ketogluteric acid plays a key role in the citric acid cycle, right? So that ammonia, see, this ammonia reacts with alpha ketogluteric acid and forms the amino acid called glutamic acid. So it forms the glutamic amino acid. So do you know the speciality of the glutamic amino acid? It shares its ammonia group. It having this ammonia group. So it is always sharing its amino group and leads to formation of another amino acids. They are almost from the glutamate itself, there is a more number of 17 amino acids are formed. 17 amino acids are formed. So once again I am telling students, Plants cannot synthesize the direct formation of nitrates and ammonia. So they are going to convert into the useful forms they are to incorporate in their body as amino acids. Amino acids. So they help in the preparation of proteins, nucleic acids, everything. Right? So now see, they have the two process, reductive amination, transamination. Got it? So when this process, always this ammonia, this is the ammonia. This ammonia reacts with the alpha ketoglutaric acid. From the cellular respiration in the citric acid cycle, you know that it is a 5 carbon compound. So it is combines with the ammonia. Then it is forms the amino acid, that is glutamic. So it is having the very special property, it is always share its amino group. So more than 17 amino acids can prepare by this glutamic. It is always shares its amino group. So when it is sharing its amino group, there is a formation of another amino acid. Got it? So this is the first process of NADH. For this reductive amination process, which is helping students, that is NADPH is helping. So I said when you are discussing the cellular respiration chapter, which means the glycolysis. So whenever the NADPH appeared, there is enzyme of dehydrogenase enzyme. As it is here forming the glutamate, so here is the enzyme is glutamate dehydrogenase enzyme. What is it, students? Glutamate dehydrogenase enzyme. So there is a release of water, and also this NADPH is reduced into NAD+. Got it? So this is the first step, reductive amination. This is the first step, students, that is reductive amination. Now see, transamination. What is meant by transferring? What is it is transferring, students? Amination, amino group. That is the transferring of the amino group. So the mostly the donor is always the glutamic amino acid. There is a glutamic amino acid is always acts as a donor. So what is it is donating, students? That is amino group is donating, right? In this process, transfer of amino group, which is transferring, students, that is amino group. So there is a transfer of amino group from amino acid to the keto group. So this amino group is transferring to the two substrates. The one substrate is the donor, right? So another substrate is the receiver. So it is accepting the amino group. So when it is accepting, it forms the another amino acid. Another amino acid along with one organic acid is also forming. See, in this process, transfer of amino group from amino acid to the keto group of keto acid takes place. Glutamic acid is the most. What I said, students, the donor. Glutamic, what is the specificity of the glutamic acid? It is always transfer its amino group. This glutamic acid is the main amino acid from which transfer of NH2, which means it's ammonia, it's ammonium compound, right? So it is always transfer its ammonia group. NH2 takes place. Thus, another amino acid is forming. So by transferring its one of the amino group to the another keto group, what is happening? There is a formation of another amino acid. At least from this glutamic acid, 17 amino acids are forming. Got it? By the formation of the transamination, this process, which means the glutamic acid transferring its amino group, no, students, that is why it is transferring, it is giving, right, transferring, that is why the process is transferring. Which is transferring? Ammonia group is transferring, that is why the process is named called as transamination, which will be catalyzed by the enzyme name called transaminase. Which is the enzyme involved here, students? Transaminase is the enzyme which is involved here. Okay, so that leads to the formation of another amino acid along with one organic acid is also formed. Got it? So there is an example like glutamic acid. Glutamic amino acid. What will form? So when it is reacts with the pyruvic acid which having the keto group, in the process of transaminase enzyme, what it will form? Alanine it will form. Alanine is the amino acid. Along with that, one organic acid formed, that is alpha ketogluteric acid. Alpha ketogluteric acid. This is also one of the best examples, students. So glutamic, this glutamic in its structure there is one, it transfer the one amino group this of the keto group of keto acid, that is pyruvic acid, and it forms the alanine amino acid. Along with that there is a formation of organic acid, that is alpha ketogluteric acid. Got it? This is what exactly the plants synthesize their amino acids, right? So this is what up to this the chapter, students. I hope you all understand this chapter. So see this, students, here is the one of the question. So see, first here there is a two process, that is direct to amination and transamination. So you have to, this is one of the two important questions, students. One is nodule formation question and nodule formation question, and also here is the how plants synthesize amino acids. These two are very important questions for your IPE point of view. So for both it is very important, students. You must and should focus these two questions. You have to be thorough with these two questions. And now see, we completed the chapter, students. Now see, let's solve first here the question. Solve this question, students. Here it is the question, that is accession. What is the given accession, students? That is free living bacteria such as rhizobium. Of course, another free living bacteria example given for the NCERT book is Frankia, right? Here is also given, students, that is Frankia. It is associated with, Frankia is the bacteria, it shows a symbiotic association with the plant of Elenus, right? With the plant of Elenus, which is the non-leguminous plant, right? So it is not leguminous plant, right? Rhizobium is always associated with leguminous plants. Leguminous plants, right? Now see, what is the given? Free living bacteria such as rhizobium and Frankia can help in biological fixation of nitrogen in the roots of angiospermic plants. Is the assertion is true or wrong? Is the statement is true or false, students? That is, it is the absolutely true, right? So free living bacteria such as rhizobium, Frankia help in the biological fixation of nitrogen in the roots of angiospermic plants is the right statement. Now see the reason. Prokaryotes exclusively, they are saying prokaryotes, which means the bacteria, right? So exclusively contain nitrogenase enzyme. So here itself the statement is absolutely correct. The prokaryotes, bacteria having this nitrogenase special enzyme which involves in the fixation of, when it is associated with the plants, it involves in the fixation of nitrogen in the absence of oxygen, right? Which can catalyze the oxidation reaction, of dinitrogen into glutamate. Here it is, what is they saying? Which catalyze, what is meant by catalyze, students? Which fast up the reaction by the oxidation reaction into dinitrogenase into glutamate. Directly it is giving that nitrogen into glutamate. Directly it is fixing in the roots. Not at all, right? So it is the wrong statement. The nitrogen is not directly giving the glutamate amino acid, right? So for the fixation of the nitrogen, it takes the 3 steps to form the ammonia. It is 3 steps to form the ammonium, right? It takes 3 steps. Got it? So now this statement is wrong. According to the question, assertion is true. But is the reason is the correct explanation of assertion? No, because the statement itself wrong. We all know that the statement is false, right? So here the first is both A and R are true and R is the correct explanation of A. This is the wrong statement. Next, both A and R are true, but R is not correct explanation of A. Here is saying both are true, but R is not the correct explanation of A. This is also wrong statement. First of all, the R is false statement and it is not at all correct explanation of the assertion. So coming to the C, A is true, but R is false. This is the right statement. This is the right statement. So this is the right statement. 1 minute. This is the right statement. Coming to the last option, just see students, A is false but R is true. A is only the right option, right? R is false. So it is also completely wrong. For the above question, students, C is the right option. C is the right option for the above question. So coming to see the question, here C is the right answer. C is the right answer for this question. See another more question, student. Students, see this question. The following are the host plants. Here they are saying that the following are the host plants. 1 minute. Where it is? Okay. The following are the host plants along with microorganisms. These are the who are placing the symbiotic association along with their particular microorganism also, which fix atmospheric nitrogen. They are fixing the microorganisms fixing the nitrogen in their nitrogen. So find out the correct match. They are saying find out the correct match. Now see the first one, Frankia. It is a free living bacteria, right? It is a free living bacteria which involves in the non-leguminous plant association. Symbiotic association takes place by the non-leguminous plant like non-leguminous plant like Alanus, right? So is the correct option. First one is the correct option. Now see rhizobium. Rhizobium alpha alpha. It is also right statement. So when we see the at the first of the class, I explained the examples now students. There is the one of the example is alpha alpha also. The rhizobium bacteria can associate with the plants like sweet clover, right? Lentils, alpha alpha also, right? This is also right statement. Alabina wheat, it is a wrong statement students. So Phosphotrium gonorrhea, it is also wrong statement. So according to the question, which are the true statements students? That is 1 and 2 are the true statements. 1 and 2 are the true statements. So this is the end of the class of today students. So we completed the chapter. So tomorrow onwards we are doing the MSET question students. Be prepared. You just skim the class of the mineral nutrition and comment your answers in the comment section while I am doing the MSET questions. Got it? EAP set questions. So if you have any doubts, please let me know in the comment section. And before I am winding up the class students, I would like to inform very important news for all of you. So here we have started the NEET long-term course, especially for the who are determined, who are passionate about the dream to become a doctor. For just Rs 3,333. You going to have benefits of live plus recording sessions. So if you do the recording and classes and also live classes, as per your convenience you can attend. If you do regularly, those portion is completing and you do the well crafted study material that we are providing, that itself can secure a very good rank in upcoming examination for sure. So you will get the well crafted study material along with the fees of this 3,333 per month students. So this is the best opportunity for who are want to become the doctor, and it is especially benefit for who are 11th and 12th college goers. So if it is a smart preparation, grab this opportunity. Very limited seats are available. So the next batch is going to start from the September 1st onwards, students. Don't miss this opportunity. And if you have any queries, let us know by this below number. It is as per your convenience. You can join online as well as offline. For online people, you can download our app name called NEET World. There we will give you access by by calling this number, we will give you access. And if you are want to join through offline, students, you can directly come to our institute name called NEET World, present in Narayani, Hyderabad. So this is about the today's class, students. So let's meet in the tomorrow class. Thank you.